Bismillahirrahmanirrahim. Welcome to Ali's Biology. In this video we will discuss about the characteristics of enzymes. Okay, number one, all enzymes they are proteins, means that if they are proteins, so they definitely like proteins, they will be made up of amino acids. Number two, enzymes they increase the rate of chemical reactions, and enzymes are not consumed during the chemical reactions. Enzymes they increase the rate of chemical reactions billions and millions times, so that more products they are being formed in a very little time. And enzymes are not consumed during the chemical reaction. It means that whenever the enzymes they initiate any chemical reaction, so what will happen, that we can say products will be formed, but along with the product the enzymes will be obtained as such as before the chemical reaction. So enzymes they are not consumed, they just activate the, we can say, the chemical reaction, increase the rate of chemical reaction, but they are not consumed themselves. Okay, then enzymes they are specific for their substrates, means that any enzyme it is specific for a specific substrate, mean that, for example, protease enzymes they will act upon only proteins, means that the proteins are their substrates on which they have to act, while they will not act on lipids or fats, they will not act on the carbohydrates. So they are substrate specific. Okay, number four, a small portion which is called active site, it is involved in catalysis, means that small portion like this. For example, I have made a diagram of an enzyme. This small portion where this substrate will get attached with it will get fit with it, this enzyme. So this small region which is responsible to recognize the substrate and bind it with itself and then convert this substrate into its products, so that will be a active site. All of the, we can say, all of the enzyme is not involved in catalysis, only a small portion which is called active site, it is responsible to recognize the substrate and bind it to itself and then convert it into its products. Okay, then next point is enzyme activity. It is being regulated by two substances, one is inhibitors and the others they are called as activators. So inhibitors they stop or decrease the enzyme activity. Sometimes we say that enzymes are we can say not necessary, so at that time the body will produce inhibitors in order to stop or decrease the enzyme activity. And then the enzymes might be, we can say, regulated by the activators, because these activators they will increase the enzyme activity. If, for example, we say that enzymes they are needed for a particular reaction, so activators will be produced by the body of an organism in order to increase the enzyme activity. Okay, then next point, some enzymes need no non-protein components for their proper function, which are called as cofactors. Most of the, we can say, enzymes they can work alone without any, we can say, cofactor, but some of the enzymes they need the non-protein components for their, we can say, proper functioning, so these are called as the cofactors. So these cofactors might be organic, they might be inorganic. So for organic we have the example of flavin and heme, and inorganic we can say these are the metal ions. So organic, we can say, organic cofactors, if they are, we can say, tightly bound to an enzyme, these 3 or 4 lines they show the, we can say, strong bonding with the enzyme. If the organic cofactor it is tightly bound to an enzyme, so such organic cofactors they are called as prosthetic groups. And if, for example, the organic cofactors they are loosely bound to an enzyme, so such we can say organic cofactors they are called as the coenzyme. I hope so that you understand. Thank you very much.